I am so excited and so honored to have the opportunity to speak to you at what is really, for me, one of the most inspirational events that I get to come to and participate in each year. So I only have 15 minutes, and I certainly don't want to stand between you and wine tasting, so I'm going to keep this quick. But I really, really welcome the opportunity to have further discussions with you later on throughout the day, so please come find me and talk to me. I really value your input. So the idea of prevention strategies in medicine, this is really not a new idea, particularly for common diseases. So the idea of, for example, screening at-risk populations, so women over 40 getting mammograms, this is really nothing new. But what's interesting is that this is actually a relatively new development in the field of mental health, the idea of early intervention. And so these are common diseases. So when you think about it, this is really a no-brainer. 1 in 4 people in this country will suffer from a mental illness at some point during their lifetime. And, we don't wait for someone to have a heart attack to say, oh, you know what, we probably should have done something about their cholesterol. So we really should not do that for brain disorders either. And this is, I think, a really astute quote from Thomas Insula, director of the National Institute of Mental Health. The way we pay for mental health today is the most expensive way possible. And I really, really think we can do better. So why study at-risk populations? Well, this is a model of illness progression for psychosis or schizophrenia. And we know that schizophrenia is a disorder that has its roots in early development. So long before the emergence of these full-blown psychotic symptoms, we see early cognitive, affective abnormalities, so abnormalities in social emotional development that are subtle. But what this suggests is that this is actually a really late stage of the illness, this emergence of full-blown symptoms. And so really what we ought to be doing is identifying people back here, intervening early, which could really make a difference for outcome and potentially prevent this whole negative cascade of events from taking place. And so that is exactly what we are trying to do. So this is just a history of our program. So in about 2002, we started the Center for Assessment and Prevention of Prodromal States, or the CAPS program, which is a program for early identification and intervention for psychosis. And this is my colleague Ty Cannon receiving the first check from the Staglins, which was sponsored by the music festival, so this very event. And this is, I don't know if you can see this, but this is what I was up to at the time. So in 2002, I decided to give my stress hormones a workout, and I moved across the country to UCLA to start a new faculty job because I really wanted to build this CAPS program from the ground up with Ty. And so then I moved. I had a baby 6 weeks later. So this is little Zach. And sometimes I feel like, gosh, nothing's really changed. We haven't made any progress. And then I know you can see this is me and little Zach now, and my daughter Kylie, and we're standing in front of the Staglin-Imro Center for Cognitive Neuroscience, our neuroimaging center that we now have at UCLA, funded by the Staglins. So progress is being made, things are growing. And so clinical high-risk paradigms, we now have a structured research interview that can very reliably identify people that are so-called prodromal, in a so-called prodromal state or clinical high risk. And so this is an interview that really identifies these early fine-grained pre-psychotic symptoms. So that and or a family history combined with a recent functional decline are criteria that predict people who are at high risk for developing a psychotic disorder, people who are at risk of imminent onset of psychosis. And so people who meet these criteria, about 30% will develop a full-blown psychotic illness within 2 years following ascertainment. And so it's not the majority of these individuals, and I really want to highlight that. But of those who do develop a psychotic disorder, about 80% of them will be a schizophrenia spectrum illness. Now, as you can imagine, this is really, really difficult to do this kind of work by yourself at a single site, to really try and gain any traction on mechanisms of illness and risk prediction. And so we formed a consortium, the North American Prodromal Longitudinal Consortium, which is a group of 8 sites who were doing, essentially had our own prodromal-focused clinical research programs across North America. And now we we've added UCSF has joined us. And so the first thing we really had to do was to take all of our differently collected sets of data and combine them into an integrated database. And this sounds incredibly boring, and it was, but it was really necessary first step for this program. And then in 2008, we started a prospective longitudinal study doing the identical protocols across all the sites: imaging, EEG, blood draws, cognition, clinical interviews. And so in doing that, we've now collected data on over 800 people that we've prospectively followed, and I'm going to tell you about two of the findings that I'm really excited about. So individual risk prediction. So when we often talk about things in terms of group averages, but if you're having symptoms and you go to see your doctor, you really don't want to hear about the group average. You want to know about your own individual prognosis. And so we teamed up with this guy here, Mike Catan, who's the head of biostatistics at the Cleveland Clinic, who's been developing risk prediction algorithms for things like recurrence of various kinds of cancer or heart disease. And so we've developed now a individual risk prediction algorithm for psychosis, and this is a web-based application. This is the link here, and so these slides will be available later, so you can take a look at it. And so if you're, so it turns out that a couple of key symptom measures, age and a couple of brief neurocognitive measures, really gave us the most traction in terms of our risk prediction. And so if you're a treatment provider and you plug in values for your individual patient, you can get a risk of their likelihood of developing a psychotic disorder within 2 years. And so we looked at this across our entire sample, and we get an overall classification accuracy of 72%. And for prediction of conversion to psychosis within a 2-year follow-up period, which is actually almost identical to the accuracy rate for prediction of recurrence of breast cancer. So a couple things that I want to point out that I think are really important here. First of all, even though we don't have an objective laboratory-based diagnostic measure for psychosis, we can actually achieve similar accuracy in terms of outcome prediction to what's seen in other medical illnesses. And the other thing is that this is something that really can be very readily implemented by a clinician in their office, on a tablet or a computer. Okay, so biomarker discovery. Koplik spots are these little tiny spots that appear about 2 to 3 days before an outbreak of measles virus. They are very, very diagnostically specific for measles. And so this search for the elusive Koplik spots for psychosis, or something that's going to be a really reliable biomarker, and that's something that we've, of course, been working on. And ideally, a biomarker will reflect something about the underlying pathophysiology, or something about the mechanism of illness. And biomarkers can facilitate treatment development in a couple of really important ways. They can provide molecular targets for intervention based on actual disease mechanisms. And wouldn't that be really nice to have something like that for a brain disorder? They also provide a fast-fail testing strategy where candidate interventions can be tested for effects on a biomarker before putting all your eggs into a large-scale clinical trial. So, on that note, I'm going to give you a 20-second overview of a brain structure called the thalamus. This is the thalamus here, this little structure. And the thalamus, we think, is really important and interesting, particularly in schizophrenia. It is basically a hub, a communication hub, through which most neural computations in the brain flow through it. And so we have these what are called corticothalamic circuits, and this kind of represents a fundamental way in which the brain is organized. And so we studied this using a technique called resting state functional MRI, which is essentially a way of looking at the brain's activity when one is at rest or not doing a particular cognitive task. And so it turns out that when you look at the connectivity or the communication of the thalamus with other brain structures in people with schizophrenia, people with established illness, we see that there's this pretty consistently identified dichotomy between overconnectivity of the thalamus with sensory brain regions and underconnectivity with structures like the prefrontal cortex, the striatum, and the cerebellum, brain regions that are involved in higher order cognition, reward, and attention. And so it kind of makes sense in terms of the the symptom picture in schizophrenia. And so the next question was, is this kind of thalamocortical disconnectivity something that actually precedes illness onset? And it turns out that it is. And in fact, we found the exact same pattern of over and under connectivity that to be predictive of conversion in those at risk. And so this is important because it shows that this brain disconnectivity, something going on in the brain, actually precedes the onset of the symptoms. Now, is this a biomarker? Well, ideally we'd like to know that this is actually somehow causally relevant to the illness, that if you fixed it, it would do something in terms of changing behavior. And, we don't yet know that. We have a lot more work to do to know whether, so really, the good news is we just got funding for the next 5 years of this project. And so for that project, one of the things we're really going to try to do is to hone in on this prodromal period, doing very dense longitudinal assessments to really try and understand what comes first. Is it something about the brain disconnectivity, or are there stress hormones, cortisol changes that precede that, or perhaps, an inflammatory process that can be measured in blood? And so the other thing that we're doing, if we thought we were going to stop at North America, no, we're not. We are now conducting a project called Harmony, which stands for Harmonization of At-Risk Multi-Site Observational Networks for Youth, which is, we took a lot of late nights coming up with that acronym, but it's pretty good, right? And so we are adopting a common protocol across not only North America, but Australia and Europe, and so across prodromal research programs. And so what you can see here that's really exciting is, this seed that was planted by the Staglins about 14 years ago has really blossomed into, something like total world domination, which is pretty cool. But seriously, we really, I think this kind of team science approach is really very critical to what we need for understanding mechanism of illness, and of course, eventually the goal of preemption of illness studies. So in the last couple of minutes, I'm just going to tell you about a new initiative we have at UCLA, which I'm really, really excited about. This is the UCLA Brain Grand Challenge, where we are focused, we're going to be over the next decade on eliminating the burden of depression in our community. And so why the focus on depression? Well, the World Health Organization has estimated that by the year 2030, depression is actually going to be the leading contributor to the global burden of disease worldwide. It's already the leading cause of disability within the United States. And depression is the largest risk factor for suicide, and it's something that touches everyone. I would imagine that probably everyone here has known someone who suffered from depression and has seen the effects of it. And depression really makes everything else worse. So the outcome, so if you have a heart attack, cancer, Parkinson's disease, if you also have depression, that recovery and from illness is much, much harder. And yet, unlike other common diseases, we really do not understand the basic biology of depression. And this is a really huge impediment to making progress. And so, so what do we need to do? Well, according to the wise words of distinguished Dr. Steve Hyman, to understand the molecular mechanisms of depression, we need to collect genetic data from more than 100,000 people. And so if Steve Hyman says collect data from more than 100,000 people, well, by golly, we are going to do it. So that is our goal over the next decade. So at the center, so really our goal here is to elucidate mechanisms of depression in order to develop new and more effective treatments. And so in that regard, at the center of this endeavor is really this 100,000 person cohort, and at the same time we'll be conducting basic science studies to really map the neural circuitry of depression. And then we're starting demonstration projects now focused on biomarkers of treatment response. And there, one of the things that's going to be really important to this effort is to harness new technology for wearable devices, watches, and phone applications that can track fluctuations in mood, activity, sleep, and then we think that these things may, and even things like voice have pattern recognition, and we think that these are things that may be able to precede and predict the onset of a mood episode. So you can see that this is really conducting team science at a whole new level, not just collaboration between academic partners in, the psychiatry, psychology, neurology, computer science, economics, but also engaging partnerships with industry, as you heard about from Dr. Manley, is going to be really important. And So, the other thing is that to do a project on this scale, it really requires thinking about the assessment of psychopathology in a really different way. And so we're planning to screen everybody in the health system in the campus of UCLA, which is about 2 million people encompassed there. So it's a pretty massive undertaking. And so we are going to be using a fairly novel methodology called computerized adaptive testing, which essentially allows you to use the information of how a person responded to one question to then inform what the next question is going to be. And it turns out that this method is actually really, really accurate in diagnosing depression and also diagnosing depression severity. So that's the goal here. And of course, my particular interest in this endeavor is in capturing those individuals with our screening who are at risk but haven't yet developed a full-blown mood episode, and then providing feedback and then stepped interventions based on illness severity, and so hopefully can change that trajectory. And so we're really lucky to have here today a lot of the really visionary leaders in the Grand Challenge: Nelson Freimer, Michelle Krask, Kelsey Martin, and so really the goal here, deploying new treatments to cut the global burden of depression in half by 2050 and eliminate it by the end of this century. Wouldn't that be incredible? And I really think that with the combined brain power, motivation, and leadership we have from everyone here today, we can really do this. So I just want to thank our funding sources who really made this work possible, and particularly the Staglin family and the music festival that really started it all. And so this is my email address here. I would love to hear from you. This is the link to the CAPS program. I also have Nelson Freimer's contact information here if you're interested in learning more about the Grand Challenge. So thank you so much for listening, and take questions.